Okay, hello. Should I share my screen? Am I already sharing?
Yes. No.
Okay. Let's do it. That way. How about that? Does that look okay?
Yeah, perfect.
Okay, awesome. Yeah, thank you very much for the offer to present today. I'm very happy to talk about the work that we do here at the OICR. Yeah, so my name's Alex Fortuna. Thank you very much for the nice introduction. I'm the manager of the Clinical Genome Interpretation Group at OICR. So this group is part of a joint genomics program. It's a collaboration between OICR and UHN here in Toronto. Together, we support a variety of clinical and research assays. Very excitingly, we recently completed our third accreditation. And so these various accreditations include the College of American Pathologists, or CAP, as well as CLIA and IQMH. So with these capabilities, we are involved in more than 10 clinical trials with the University Health Network and different partners in the United States. So as a clinically accredited lab, we have a variety of different offerings within our program. So these include different options for whole genome and transcriptome assays at various depths, as well as targeted sequencing panels, and the option to include shallow whole genome sequencing. As an accredited lab, we currently accept fresh frozen tissue or FFPE and blood. For our whole genome assays, we currently have a turnaround time of 45 calendar days. And for targeted sequencing, we have a turnaround time of 21 calendar days. And we are always actively working to reduce this time. So as we provide both whole genome and targeted sequencing assays as options to our community stakeholders, it's important to note the distinction between the two samples, or when one may choose one over the other. So a whole genome covers all genes and intergenic regions, and it gives improved copy number and structural variant calling. This is a lower coverage, and it comes at a higher cost. Alternatively, we offer targeted sequencing. So this is where we enrich for a few cancer genes, but it's not uncommon to say have a panel of 500 genes or so. This is especially interesting as it has a high sensitivity for low variant allele frequency mutations. This is a much higher coverage. So as you saw on the previous slide, it's often say 10 to 15,000 X, and it's at a much lower cost than a whole genome. Interestingly, one approach may be to order a whole genome, and then if a clinician is interested, they could interrogate a smaller portion of the genome by ordering targeted sequencing. Also, as this is a bit of a change from the standard of care, which may often include panels, we have a vision to basically use the complete genome and transcriptome sequencing to aid in precision medicine. And in this way, I believe that there are some distinct advantages over some mutation and fusion panels which are currently offered. With whole genome, we can more accurately estimate TMB, and we can more accurately determine tumor mutational signatures, as both of these elements benefit from having more data. We can potentially identify novel oncogenes, as these are not limited to what's currently known as we're interrogating the entire genome. And with the whole transcriptome, as we have as we can view expression for many genes, we can potentially characterize the immune landscape as well as identify novel fusions which may not be present on a fusion panel. And often we look to incorporate these two assays together, such as when looking at a candidate fusion, we can look for support in the genome. So just an example of when this process may be utilized. We could look at various samples over time, and from this, we can then potentially recommend a treatment. So for lung cancer, one of the most common mutations is EGFR, and it's an important marker for treatment decisions. Thus, if we're able to track the mutations over time, we can we can make evolving recommendations based on the tumor's profile. So for the rest of the talk today, I'm hoping to review the different components of our accredited process, from sample submission through to reporting. So the first step in our process is the requisition system. So a requisitioner is one who's in charge of the sample submission. We've developed a portal where a requisitioner who's in charge of the data control for a study can enter and order these tests through an online system. So as a bioinformatician, we do not have any access to PHI. This is a very important component of our process. So they provide us with de-identified information, but we do get information which we require for our interpretation, such as the cancer type. And sometimes we'll get information such as the results of previous genetic testing. And once it goes through the lab and the various interpreters, we upload a de-identified clinical report, which again, I'll get into a little bit later. And then we work with a clinical medical geneticist at OICR. This is Dr. Trevor Pugh. He reviews the report and will sign it and add the PHI back on, where then it is then uploaded to the requisition system, and the requisitioner can pull it down and use it to potentially inform treatment. So once the sample has been received and extraction has been performed through Tissue Portal, OICR does the sequencing, and this kicks off the exciting informatics process. So here you can see just a snapshot of the tools which we are using to collect information for our clinical reports. We affectionately refer to them as the blue boxes. So this is the output of various pipelines which we run, which you can see in yellow here. So on the left is the whole genome sequencing pipeline, where the outputs are somatic copy number variants, somatic single nucleotide variants, indels, and the tumor mutation burden, and structural variants. And from the whole transcriptome sequencing, we are mainly interrogating fusions. Right now, our two popular colors are Ariba and Star Fusion. And we also get information on an expression profile, which we currently are using a tool called CyberSort. And from here, we are beginning to perform some immune inference. And last but not least, all these blue boxes are fed into a tool called Gerba, which I will certainly get into a bit later. But before I get too far ahead of myself, the first and one of the most important processes is performing quality control. We use an online portal based on the Python Dash framework. At OICR, we call it Dashi. We have a soup theme for some of our tools. So in this way, we perform pre-analytical sequencing from the Illumina MiSeq and full-depth sequencing on the Illumina NovaSeq. And importantly, this allows us to both catch bad samples and see trends in library results over time. Some of the important metrics which we're looking at include coverage, duplication rate, as well as insert size. As we process many samples here, having such a nice framework for review has been instrumental in allowing us to stick to our tight turnaround time. Interestingly, another important quality metric is tumor purity. Currently, tumor purity is estimated using a tool called Sequenza. It provides us with a variety of solutions, and that includes estimating the cellularity. In this case, estimated cellularity is 58%, and the ploidy is 2.8. This is the same tool that we use to estimate copy number variation. The plot shown on the right is an example plot from Sequenza. Here we can see the y-axis is the copy number, the x-axis is the chromosomes. So we can see that this is a rather noisy genome with a ploidy of 2.8. And this is an example case which I will get to at the end of my talk. But you can see how the tool is able to estimate both copy number gains, which are shown in red, and some of the copy losses, which are shown in blue. So once we have established that the sample meets the quality threshold, we then are able to continue with our interpretation. Gerba collects input from a variety of sources into a tidy JSON file, which we can use for both reporting and long-term storage via a CouchDB database. A very important step of Gerba is the annotation with OncoKB. This allows us to identify oncogenic mutations. It collects metrics from our LIMS system, quality control metrics from Dashi, so information such as coverage, which will go on the clinical report, and information from the requisition system, such as cancer type and the OncoTree code. So OncoKB is a precision oncology knowledge base developed by MSK. Those of you who may have used cBioportal in the past may be familiar with their annotation system. It is the same engine, it's the same developers. So OncoKB basically distills FDA and NCCN recommendations into an API, which we can query with the results of our clinical pipeline. In addition to those drugs with an approved treatment, it also annotates mutations which are oncogenic or likely oncogenic. These decisions are made by a team of geneticists who are constantly reviewing different papers related to the field related to the field of cancer genetics, and they provide updates regularly, and they have quite a good guide explaining their logic for the different interpretations and assigning them to the various levels. So as I mentioned earlier, the output from JSON is this, the output from Jerba is this JSON file, and we then have to translate it into something that's a bit more human readable, so that it can be both reviewed by us and it's informative to the clinician. So right now we use the templating language Maco to turn the JSON file into an HTML, and finally into a PDF where one of us can look at it. So once we have the HTML, we review each of the different sections. This includes all of the candidate somatic mutations, copy number events, and fusions, and every single event is reviewed in order to be included on the final report. So we review each of these mutations in IGV, the Integrated Genome Viewer. Perhaps many of you are familiar with it. So most of the time, these are good calls. So this is an example of a large deletion insertion in EGFR. There's no issues here, and this mutation would then make it onto our clinical report. The odd time will open a IGV link, and we'll see some evidence of perhaps a miscall. This could be to poorly mapped reads or to a noisy area of the genome. And if we're not satisfied with the call and there are some detailed elements in our data review SOP, this mutation will be removed and not included on the clinical report. In a very similar manner, we perform review on IGV for fusion. So we look at the breakpoints and we view the spanning reads. So it's a bit more interesting with IGV. You can paste the candidate breakpoints and it'll do a split read view where you can see the reads on either side of the breakpoint. And if you hide the, or if you show the soft clipped reads, you'll be able to, it's actually quite interesting. You'll be able to see that those soft clipped reads, if it's a spanning read, actually will read into the other chromosome. So if we see this evidence that is support that we are seeing a high quality call. In cancer, when there is a oncogenic or driver fusion, we frequently see that there is quite a bit of evidence. So it's rather clear to us that it's a positive call. Sometimes you'll see a very few spanning reads, and that's when we might call the fusion into question, or alternatively, if that read may map to various places in the genome, it may not be a good call. So as it's a clinical report, there are some sections which we are mandated to include, and this allows us to keep our CAP accreditation. So we need information about the testing laboratory, we need information about the patient sample, and we need some information about the test. We also need information about the genes tested and the limit of detection. And then when it comes to variant and result information, we need information about the actual variant and the relevance to the variant or the condition tested. known responses to therapy or prognostic information, as well as evidence supporting classification of the variant. So now I'll just go through a bit of an example of a report which we've recently completed. So at the top, we can see some basic information about our testing laboratory, as well as some quality control information. So at the top is our header with our lab information. The assay, this is the assay which they ordered, which was selected through the requisition system. The cancer type, and the biopsy is also something that's entered through the requisition system. And as we can see here, we never have access to any PHI. So things like the patient name and the date of birth, physician is all left blank. That only our clinical medical geneticist has access to that information. And then sample information. So we have our OncoKB code, which is a helpful indicator when doing annotation of a specific variant, because OncoKB will have different classifications for any given variant by its cancer type. A coverage, so it's 85, so this is above our 80x threshold, which I mentioned earlier, and the estimated cell content being 58%. There's a section here on actionable mutations. So unfortunately, this case didn't have any actionable mutations, but if they did, this is where they would go. And a summary of results. So this is where a lot of the clinical genome interpreter's time would go into, especially if we're looking at a new cancer type. We spend some time reviewing the literature. We found there to be a helpful website called My Cancer Genome, which can provide us a list of the most frequently mutations in each cancer type. And we also highlight here we highlight any mutations which we observe. So though there are no actionable mutations, we did see several mutations which are commonly altered in a high-grade serous ovarian cancer. So TP53 point mutation, as well as amplifications of PIK3CA and KRAS. And we comment on the ploidy as well as the tumor mutation burden. So the Genomics Statement, as I just said, it's an opportunity for us to highlight any key features which we want to make sure that the clinician or anyone reading the test is aware about. So it's important for us to provide context. So we say that they've been diagnosed with a given cancer type and they've been referred for a particular assay. Then, if applicable, we highlight any potential treatments, comment on any key genomic landscape findings. This may include the tumor mutation burden or the ploidy, and really any other insights which we may have. And these are the other sections of our clinical report. It's a bit hard to show the report on a slideshow, but I promise it doesn't look half bad as a PDF. We have a genomic landscape section. So we highlight the TMB. The red dotted line indicates the cutoff for TMB high, and the black line indicates this tumor. So as we can see, though the tumor mutation burden is high, placing it in the tumor mutation in the 53rd percentile, it's not high enough to say qualify for some particular TMB-based treatments. We have a small mutation section. So this may be interesting. It's a plot of the variant allele frequency. So this shows the distribution of VAFs across the tumor. So there's a peak around the 20 to 25% range. And then we have the listed genes here, which we observe. So although we see over 2,500 somatic mutations, and 117 of these impact coding sequences, only 2 correspond to oncogenic mutations as defined by OncoKB. And here they are, and these are actually hyperlinks, which will bring you to the corresponding OncoKB page. So it's a good resource for a clinician who wants to learn more about the particular variant. And lastly, we have a section on copy number variation. The percent genome altered was 60%. We define percent genome altered as any section of the genome where we are seeing evidence of a copy number gain or loss, which meets our specific threshold. And we have a copy number plot. This is actually a rather new addition for us, but I think it can highlight to a clinician whether or not a group of copy number amplifications or deletions are co-occurring, which is something that people are commonly interested in. Yeah, and last we have a section on our assay description. So we go into more detail about the OncoKB level and the definition. And we provide some more information about the other definitions, which may be on a report, callability, coverage, etc. And last there, we provide information about the specific version of the tools which we are using. Yeah, so that's our report to date. We have lots of stuff in the works for the near future. We're very excited about adding especially information related to mutational signatures on our clinical report. This is something that a lot of trials are interested in. Very soon we'll be adding a microsatellite instability score to our clinical report, as well as some information on the expression of different genes. And hopefully early next year or in 2023, we'll be adding HRD to our report. As again, this is something that the community has been very interested in having. So with that, I would like to thank my team very much. They've been instrumental in this work. Both Ian and Felix completed a lot of the work which I showed you today. Gerba was Ian's big project, and Ian and Felix has been working hard on the both the mutational signatures and a large report redesign, which is the report which you saw today. And I'll also note that my group is hiring. So if you're interested, we're hiring a bioinformatician. So you can reach out to this to me through the email listed here, or we have a job posting on the OICR website. So with that, thank you very much for listening today, and I would be more than happy to take any questions. Thank you.
Thank you, Alexander, for your presentation. That was very interesting as presentation, and I learned a lot about the interpretation of variation. I have a question, and I let's see if some of attendees have questions. No? Okay. So I'm just wondering, how do you do the filtration of variant? Because you didn't talk about it.
Yeah, so we call magnetations now with mutate2. we have a VAF threshold, which we've set through our limit of detection. So it needs to have a VAF greater than 10%. And then we also do the filtration by eye using IGV, so double checking that the call was made correctly. And then, though there may be thousands of mutations called across the genome, only those which are annotated by OncoKB will actually make it onto the clinical report.
So I as understand that OncoKB, he does also the filtration of the variant, which is not really one.
Well, not so much filtering, but more the annotating. Like we do the filtering ourselves. So, I mean, we do tumor normal calling. So we don't have to do, like if we were doing tumor only calling, like sometimes we'll filter by like a panel of normals or exact and nomad, that kind of thing. But like tumor normal calling with mutect accomplishes much of that for us.
I see. And how much like you have variant per sample?
Sorry?
In general, like you have how much you get variant in total? Like, do you have 100 variant? I'm just curious.
I mean, on a clinical report, there's very few. I think the example that I showed is pretty representative of most reports. Like there are, there's usually just a handful of somatic oncogenic variants, unless it's a particular case, which is like a hypermutator, in which case we'd get thousands of variants. But in a cancer genome, I mean, I think, it varies per cancer type, but usually there's a tumor mutation burden between, 1 and 3, which is representative of a certain number of mutations. But it varies a lot by cancer type, which is why we have a tumor mutation burden percentile based on like a cohort.
Nice. And do you use other database on the annotation?
For oncogenicity, no, not right now. We want to. We're certainly going to get into it. I mean, because it's a clinical report, we're trying to limit ourselves to what's appropriate, which is often what has been FDA or NCCN approved. But a lot of people are looking for some more specific research stuff, so we could start to explore those databases, but we're trying to limit our scope a bit as the clinical program. Did Matthew have a question?
Yes, sorry. Yeah, so in fact, Yasmin just asked it, I guess. I was wondering about OncoKB and what motivates the choice of that particular database, let's say compared to Civic or CGI or I mean, other knowledge base that you may find maybe other variants there that they are not reported in OncoKB, let's say.
Yeah, I mean, we have a bit of a working relationship with MSK because, like, traditionally before we had a clinical program, we would upload our projects to CBio portal. So I guess, it's the one we're most familiar with. And then we found the API very easy for us to work with. So there's a MAF annotator, a CNV annotator, Fusion annotator, so it's actually really quite nice. And we like that it was a controlled environment with just a few medical geneticists are able to make changes. I think with Civic, it's a community annotation, so maybe we didn't want something that was quite as malleable, I guess. I mean, we definitely are. We're also kind of a newer program, so we just kind of hit our second year. So I think now that we have an established workflow, I think we are interested to see it. And if you have any suggestions, let me know.
Yeah. I know it's a lot of work to try to make work together different data source. That's for sure, using probably different evidence level or ontologies for disease and maybe even like for the variant description. You also talk about you're not integrating right now gene expression analysis, right? So you're doing the whole transcriptome. So it's a way for you to maybe help in identifying conflict number and fusion.
Yeah, so we're doing the whole transcriptome now. We use Birba and Star to look for fusions, and then we are running RSEM to look at expression, which we then provide to CyberSort, which will do an immune inference component. I think like one of the reasons we don't have an expression on a report right now is because expression's not something where there are approved treatments. So, and it's also not often, it's not directly annotated as oncogenicity. So though an amplification is annotated as oncogenic, if we had a overexpression, we don't currently have a way to annotate it, but we actually are doing all that work on the back end, and we're kind of just waiting on how to best put it on the report. But I mean, I always thought that we would use expression as evidence for an amplification or a deletion, but it's not always so linear, I guess.
Okay, it's cool to see that so you're able to orient sometime also therapeutic choice, right? So that you from time to time. So the last example you provide, there were no significant or actionable variant discovered for that patient, but from time to time you can let's say orient the prognostic or even maybe the therapeutic choice, right?
Yeah, I probably picked a bad example. Recently, we've actually had several cases of like actionable oncogenic fusions, which I think were really interesting, and they can definitely guide treatment. And I think that's a nice thing with the transcriptome is we can find those fusions, and then more times than not, if it's an oncogenic fusion, we can also find the structural variant. So more evidence that that's what's happening.
Is there? Do you include also? I mean, clinical trials into the analysis. Do you kind of look for available trials, let's say, in regards to the variant that you just find out?
Not directly right now. I know it's something that we're working on. I think in the States, there's a nice API for clinical trial matching. For Canada, I think Trevor Pugh, our director's lab, is working on something like that. But it's more often the project is part of a trial, and the sponsor may send someone to us to evaluate certain variants for inclusion, like in the trial that way.
Okay, so interesting. So you, what's your criteria to be admissible, let's say, to this program? Because I wonder, it's probably like restrict to a kind of small amount of people, or what's your let's say your ability to treat a higher load of patients right now?
Oh, I mean, it's not like I think we can support a higher load. I think it's more. I mean, like we're doing a lot of work right now with Terry Fox Marathon of Hope. Like I know, like traditionally back in the day, like I was part of a project where I feel like often we would review cases which are non-responders or people who aren't responding to first or second line therapies. But that relationship of how someone gets their genome submitted to me is actually a bit out of my grasp. I'm just the interpreter.
Yeah, sure.
I think it's a program question.
Okay. Hey, thanks a lot, Alexander. Answers a lot of questions for me, and I appreciate a lot of your talk tonight. Thanks.
Oh yeah, happy to chat more anytime. Nice to meet you.
I don't know if we have more questions. If not, I have a last question about immunoexpression. I'm just curious, what do you do exactly, or what do you mean by immunoexpression?
Oh, well, there are some tools. So, well, one example is we're working as part of a clinical trial right now where, there are a certain class of drugs which can be recommended to people with a immunogenically active tumor. So CyberSort is one tool which will estimate the population fraction of immune cells. So there are some particular immune cells which are which we believe to be evident of we call it like immunogenically active tumor, CD8 positive T cells. So if you have a certain
live streaming is on.
Oh, let me see. I mean, we looked at lots of different packages when we were deciding what to do. I haven't heard of this or looked at this in a bit. This actually looks pretty interesting. I will take a look at it.
All right.
Cool. It's got everything. There you go.
Yeah, exactly. It's pretty interesting. That's what I was wondering. But anyway, thanks. Thanks for the talk again.
Oh, thank you.
Perfect. So we're at the end of the meeting. Thank you so much, Alexander, for your presentation. MonBUG wish you good holidays for the end of this year, and see you next year for a lot of presentation and meetup coming soon. So thank you so much. And have a good day.
Thank you. Have a nice day. Bye.
Pedro.
An accident. Thank you.